Second messengers. Dun, 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 dun. Joke lang. Second messengers are intracellular substances, nasa loob ng cell, that mediate cell activity by relaying a signal from an extracellular molecule bound to the cell surface. So, ano ulit? So, kung may hormone, okay, draw natin. Ito yung cell mo. Ito yung receptor. Yung hormone nag-bind sa receptor. Okay, may ma-activate dyan. Okay, depende pa sa hormone. Then, may effector dito. Si effector magluluto ng second messengers. So, yung mga yan na nagawa intracellularly and taga-mediate ng cell activity, taga-utos, taga-send ng PM or group message, i-relay yung signal from an extracellular. Anong gusto mo, boss? Sige, i-relay ko. Okay? Ito yung first messenger, ito yung second messenger. There are various types of second messenger. May cyclic AMP, may IP3, may DAG, etc. Okay? It all depends here kung sino hormone ka. So, G-protein and adenyl cyclase. So, si G-protein, pwedeng GS or GI. Bakit red? Ayoko ng red. Okay, so once a hormone binds to the receptor, again, kung GS to, stimulatory, G-alpha ma-activate once GTP is bound to this subunit. Si G-alpha i-activate si adenyl cyclase, letter C, luluto ng cyclic AMP na letter C din. Then, it will activate letter P, protein kinase, mang-phosphorylate ng stuff hanggang masunod yung gusto niya. Okay, kapag yung hormone naman na yan, umupo sa GI, no? GI. So, receptor na bound sa inhibitory na G-protein. So, yung alpha subunit niya will do everything to shut it off. Adenyl cyclase won't be activated. Wala kang cyclic AMP. So, pang-inhibit. Kaya nga inhibitory. Okay? Yun Tagalog, Doc. O sige. A very common downstream effector of G proteins is adenyl cyclase, which facilitates the conversion of ATP to cyclic AMP. When a ligand binds to a receptor that interacts with a G protein composed of an alpha subunit of the alpha S class, adenyl cyclase is activated, thereby increasing cyclic AMP levels, and as a result, activating protein kinase A. By phosphorylating specific serine and threonine residues on proteins, protein kinase A regulates effector protein activity. In contrast, when a ligand binds to a receptor that interacts with a G protein composed of an alpha subunit that is alpha I or inhibitory class, adenyl cyclase is inhibited and thereby reducing cyclic AMP and consequently reducing protein kinase A levels. Cyclic AMP also regulates some effector proteins directly, such as ion-gated channels. Ano pa? Cyclic AMP, pagtapos na yung trabaho niya, will be degraded to, hindi na nakasulat dito, to AMP, adenosine monophosphate. Pag tapos na yung trabaho niya, no? Ayaw mo naman forever naka-turn on yan. Sino magde-degrade? This enzyme, phosphodiesterase. Okay? Yung enzyme na yan, binablock ng certain drugs, no? Yung caffeine, ano pa, yung mga methylxanthine. Kaya pag nakakape ka, inhibited si PDE, ang haba ng buhay ng CAMP. So, ayan. Okay? Caffeine can prolong a cellular response mediated by CAMP and protein kinase A. In addition to signaling in the cytoplasm, the catalytic activity of protein kinase A can enter the nucleus of cells and phosphorylate and activate some transcription factors. Okay? So, CAMP is the most common second messenger system. Okay, again, alpha subunit. The hormone binds to the G-protein coupled receptor. Ito yung receptor. Andito yung G-protein. Pag na-activate na si G-alpha, kasi may pera na, punta siya sa effector. This is an enzyme. In this case, kapag cyclic AMP kasi, ang magluluto sa kanya ay si adenyl cyclase. ATP will be converted to cyclic AMP. Letter P yan. I-activate niya to. Si protein kinase. Kinase, pang-phosphorylate ng stuff until masunod yung cell response. Know the sequence na tanongin sa exams. Okay? And then, pagtapos na yung trabaho, si G-alpha magiging bound to GDP naman. And yung cyclic AMP, degrade na ni PDE. Cyclic AMP will become 5' AMP para naka-turn off na siya. Xanthines, ano pa? Kape, methylxanthines will block this para prolonged ang effect ni CAMP. Okay, so hyper ka. This is just a sequence. Sinabi ko na yan paulit-ulit. Okay? Dok, ayaw makinig sa'yo. O, di ito, panoorin mo. Walang audio. So, ako na lang. So, hormone dumikit, G protein gets activated. Okay, GTP dumikit, G-alpha lipad, punta sa effector. In this case, it's adenyl cyclase. Pag yan yung primary effector, cyclic AMP ang maluluto niyang second messenger. Maraming cyclic AMP ang magagawa. Oh, yan o, marami. See? Galing siya sa ATP, Letter P yan. I-activate ang letter P din. It's protein kinase A. Dumikit sa regulatory subunit ng pKa. Ma-activate si protein kinase A. Tumigil na. Then, mag-phosphorylate siya. Okay, ng mga proteins. Phosphodiesterase, PDE, ng de-degrade. Okay, ng de-degrade lang siya. Kung ito yung second messenger mo, i-degrade din niya to GMP. Okay? Some hormones kasi nagbabind din sa GPCR pero ito yung naluluto. Instead na adenyl cyclase yung effector, guanylyl cyclase. Okay? So, pag gumawa ka niyan, ganun din, protein kinase G naman yung ma-activate, etc. etc. Pag ayaw mo na yan, i-degrade mo. Okay? Patawag mo si phosphodiesterase to form this one. Pag nag-decrease yung cyclic GMP sa mata mo, it leads to closure of cGMP's cation channel, leading to change in membrane voltage. G proteins also regulate phototransduction. Nice to know muna. Pagdating nyo pa to sa neuro, specifically sa vision. In rod cells in the eye, absorption of light by rhodopsin activates the G protein transducin, which via the alpha T or alpha transducin subunit activates PDE, phosphodiesterase. Activation of this phosphodiesterase lowers the concentration of cyclic GMP and thereby closes a cyclic GMP activated cation channel. The ensuing change in cation channel activity alters the membrane voltage. The exquisite sensitivity of rods to light rods can detect a single photon of light due to the abundance of rhodopsin in rods and amplification of the signal. Nosebleed na doc. Huwag mo na intindihin. Okay? Basta the thing is, sa mata, eto, napopromote yung degradation. Okay? Yung pagkonte ni cyclic GMP. Again, CAMP is the major, ang pinakamaraming ginagamit na second messenger. Okay? Different peptide hormones can either stimulate or inhibit the production of cyclic AMP. Ito yung mga sa GS and ang second messenger ay cyclic AMP. Huwag mo memorize, ang dami. Okay? Ito na yung mga GI, mga kontrabida. Alpha-2, angiotensin 2, soma-2 statin, acetylcholine. O, di ba? Madali i-memorize. GI ang inuupuan nila. Okay? So, ATP to cyclic AMP, dun siya nanggaling Enzyme that made it, it's adenyl cyclase. Pwede mong ma-stimulate si enzyme, okay? Stimulate or inhibited by different peptide hormones. Ang receptor or upuan, it's either stimulatory or inhibitory din. Ang G protein ng kasama, stimulatory or inhibitory din. Pareho may alpha, beta, and gamma subunits. Okay? Ang alpha subunit, yan yung may G, DP, or GTP. Okay? GTP. Ang beta gamma, wala. Naka-ano lang. Pa-chill-chill lang siya dun sa membrane. Naka-connect. Pag ayaw mo na kay CAMP, ipa-chop-chop mo na kay PDE, phosphodiesterase. Okay? So, ito yung cell mo. Ito yung receptor. Ito yung G protein. Pag nag-bind si hormone sa receptor na stimulatory, DGS yung ma-activate, no? Pag nag-bind sa receptor na inhibitory, tulad nito, umupo sa mga inhibitory yan, GI yung ma-activate. Okay? So, entails the exchange of GDP by, ito na yung papalit, and concomitant dissociation of beta-gamma from alpha subunit. Okay? So, yung cyclic AMP na yan, inaapektuhan ni cholera and pertussis toxin. Si cyclic AMP, ang pinopromote niya is yung active form, yung G-alpha stimulatory, which has intrinsic GTPase activity. GTPase yung taga-degrade, no, ng GTP papuntang GDP. Yan. Okay? Pag active pa, naka-on pa, GTP nakadikit. Pag inactive, GDP. Okay? Yan. Siyempre, ayaw mo naman forever naka-active, di ba? Naka-activate. So, clinical application, cholera and pertussis toxin. Bakit? Yan. So, since the alpha subunit in GS differs from that in GI, the proteins which are distinct gene products are designated as alpha S and alpha I. The alpha subunits bind guanine nucleotides. The beta-gamma subunits are likely always associated and appear to function predominantly as a heterodimer. Wala lang. Ganda lang yung ambag niya, kumbaga ganun. The binding of a hormone to RS or RI results in a receptor-mediated activation of G protein, which entails the exchange of pera, yung GDP maging GTP, on alpha subunit and the concomitant dissociation of beta-gamma from alpha. The alpha S protein has intrinsic GTPase activity. The active form, alpha S na may GTP, is turned off or inactivated on hydrolysis of the GTP. The trimeric GS complex alpha beta gamma is then reformed and is ready for another cycle of activation. Cholera and pertussis toxins catalyze the ADP ribosylation. Tinatanong sa boards yan. ADP ribosylation of alpha S and alpha I subunits. Okay? Pinupukpok niya ng ribose, yung alpha S and yung alpha I. Si cholera, alpha S yung pinapakialaman. Pinupukpok niya ng ADP ribose, si alpha S. Si pertussis, may I, alpha I, yung pinukpok niya ng ribose na may ADP. So, what, doc, pag pinukpok niya, ano mangyayari? Oh. In the case of alpha S, pag napukpok siya, this modification disrupts the intrinsic GTPase activity. Wala kang GTPase, so yung alpha S mo, forever na nakadikit sa GTP. Kasi wala si GTPase. Ayan o, wala to. Oh, ang trabaho kasi ni GTPase, i-convert si GTP papuntang GDP. Uwian na. O ngayon, hindi siya makauwi kasi pinukpok si cholera, pinukpok itong alpha S, it will turn off GTPase activity. Forever kang ganto. Si alpha S, hindi siya makauwi sa brothers niya. Okay? May problema yan kasi alpha S, isa sa mga effects yan, i-activate yung mga channels eh. Lalabas yung chloride, etc. Magtatai ka dyan sa cholera. Anyway, pertussis, ang pinupukpok niya, ADP ribosylation of alpha I. Tandaan na, inhibitory. So what? O ano ngayon? Anong mangyayari? ADP ribosylation of alpha I prevents the dissociation of alpha I from beta gamma and free alpha I, thus cannot be reformed. This activity in such cells therefore unopposed. Itong inhibitory, pinukpok mo. Oh, hindi naman siya makapagsarili. Yun yung problema. Lagi siyang andito sa poder ng ano niya, ng mga siblings niya. Okay? So pag hindi siya makagora, walang magi-inhibit sa adenylyl cyclase mo. Ganun lang naman to. Walang kukontra dun sa kapatid nilang stimulatory. So, ang dami mong sinabi, Doc. Ano ba talaga? Si cholera, in a nutshell, ayaw makipagbalikan. Forever single. Ito. So, forever turned on. Si pertussis, yung alpha I ang concern niya. Yung alpha I, ayaw makipaghiwalay. Okay? So, hindi siya makapagtrabaho. Hindi niya mapakalma to. Kaya nga unopposed activity. O ganun din. Okay? So, ayaw umuwi ni GS. Lagi naka-turn on. Tingnan mo yung cholera. Forever naka-turn on. Okay? Ayun, no? ADP ribosylation. Pinukpok. O, tapos, si alpha S mo, hindi na siya makauwi. Forever siyang ganto. Namimiss ko na yung siblings ko. Gusto ko na magpahinga. Gusto ko na umuwi. Hindi. Magtrabaho ka na magtrabaho. Isa sa mga trabaho niya, yung pagbukas ng channels eh. So, lalabas ang mga ions, sodium, etc. Where sodium goes, water follow. Magtatai yan. Something like that. Okay. Yan. Kapag si cholera mo, inapektuhan si GS alpha or si alpha S, ano mangyayari? Forever siyang turned on, di siya makauwi sa siblings niya. O di forever turned on din to, forever ka may cyclic AMP. Isa sa mga pupukpukin nito, ito turn on niya yung CFTR channel which pumps chloride out. Sino bang partner ni chloride? Si sodium. Pinaprint out. Where sodium goes, water follows. So, andito kayong lahat sa lumen. O, di mag-watery stools ka. Rice watery stool ang description ng cholera. Ito naman, walang pampatay. Wala si GI. So, laging naka-on. Okay? Yan. Ito si inhibitory, oh. Kailangan mo rin si alpha I, syempre, para mag-inhibit yung paggawa ng cyclic AMP. Ang problema sa pertussis, alpha I, shut up ka na. O, di forever to. Okay? G proteins also regulate phospholipases, a family of enzymes that modulate a variety of signaling pathways. Ligands that activate receptors that are coupled to alpha Q subunits stimulate phospholipase C, di ba? QC. Yan. Ito yung counterpart ni adenylyl cyclase. This is an enzyme that converts phosphatidylinositol 4,5-biphosphate, nice to know na yan, basta PIP2, nananahimik na phospholipid sa cell membrane, kinatay, okay, into IP3 and DAG. IP3 is inositol triphosphate. Okay? So, ayan na. Tapos may kanya-kanyang trabaho tong mga second messenger na to. IP3 is a second messenger that diffuses to the endoplasmic reticulum. Kakausapin niya to. Uy, labas mo naman si calcium mo. Yan. So, it activates a ligand-activated calcium channel to release calcium into the cytosol, whereas DAG activates protein kinase C. C ha? Protein kinase C. Okay, GQ, phospholipase C, IP3, DAGI, PKC. Yan. Hindi protein kinase A, kasi protein kinase A sa cyclic AMP yan. PKC, it's a kinase, it will phosphorylate stuff. O, yun na. Hanggang masunod yung gusto ng hormone na yan. Okay? So, PKC phosphorylates effector proteins. As noted earlier, both calcium and PKC influence effector proteins as well as other signaling pathways to elicit responses. So, as with yung cyclic AMP kanina, memorize the sequence. Okay? Si hormone, magbabind. Sinong hormone yan, Doc? May RH sa pangalan usually, ha? Growth hormone-releasing hormone, thyrotropin-releasing hormone. So, yung mga may RH except CRH, corticotropin-releasing hormone. So, pag nag-bind si hormone dyan, sa receptor, ma-activate si G-protein, magiging GTP ang nakabind sa alpha subunit. Activate niya si phospholipase C effector system. Phospholipase C, gagawa ng IP3 and DAG from nananahimik ng phospholipid sa cell membrane. IP3, may number, it will kausap endoplasmic reticulum to release calcium to the cytosol. DAG, walang number, it will kausap or activate protein kinase C pang phosphorylate ng stuff. Calcium will also help activation of protein kinase C. Okay? May other functions pa si DAG na nice to know muna for now. So, know the sequence. Ito yung mga second messenger natin, si IP3 and DAG. Okay, so ano ba talaga si phospholipase C, Doc? Kung may phospholipid ka, yung may head and tail, phospholipase C will cleave here. Okay? Puputulin niya yan. Pag naputol yan, magiging IP3 and DAG. Yung isa, IP3, yung isa, DAG. Okay? Nice to know na yung structures. Kasi fisyo tayo. Okay, si phospholipase ito yung kakat niya. So, si hormone nag-bind sa G-protein coupled receptor. In this case, it's GQ. Okay, ang bagal. Na-activate si G-alpha. Si G-alpha ang lilipad papunta dun sa effector. In this case, it's phospholipase C. Phospholipase C will cleave yung phospholipid dyan into IP3 and DAG. Yan. Si IP3 at si DAG. IP3, siya ba yung sa calcium o hindi? May number, oh. Siya yung sa calcium 2+. Si DAG naman, siya yung mag-activate ng protein kinase C. Ba't ako nag-annotate? Alam ko may audio to.
As heads and tails. The head part contains a phosphate group, and the tail is made up of fatty acyl chain. So basically, phospholipase C gamma separates the heads and the tails of these phospholipids molecules. So we have inositol triphosphate, which is the head part, and the diacylglycerol, which is the tail part. What do they do? Well, both IP3 and DAG activate a protein called PKC. PKC, or protein kinase C, is a kinase that phosphorylates a number of important proteins that then go on to influence various different cellular processes. Now, inositol triphosphate, or IP3, also has another important function, and what that is that it actually goes on and activates vesicles that contain or store calcium within the cell. By activating them, it causes release of the intracellular calcium storage. So the intracellular calcium levels go up, And when intracellular calcium levels go up, they also cause a number of changes that affect cellular processes and functions.
Okay. Ion channel link, signal transduction pathway. Wala. Pag nag-bind si ligand, bubukas ang ion channel. O, lusot ka na ion dito sa channel na to. Hindi ion ni vice ganda ha. Ion, yung positive ion, negative ion. Okay? Yan. So, again, pag napasukan ka ng something na positive, ikaw ay nade-depolarize, yung membrane potential mo mas nagiging positive. Okay? Pag mas negative ka na sa loob compared sa labas, na hyperpolarize ka. Protein kinase, wala. Kinase ng phosphorylate ng stuff. Pag pinukpok kasi yung mga proteins, yung iba na-turn on, yung iba na-turn off. Phosphorylation is adding a phosphate group to a protein or other organic molecule. Usually, it will turn on an enzyme. Yung iba, turn off. Yung calcium na naluto, okay, from or pinalabas ni IP3 and DAG, ginagamit yan ng certain molecules. For instance, ito si CAM, si calmodulin. Calmodulin is needed for for movement. It's always associated with muscle and movement. So without calcium, hindi ka magko-contract ng muscle. Gusto mo ba yun? Hindi. Okay, wala lang, sinabi lang dito. But important yung calcium na yan. Okay? So it's for contraction. Yan. Pag tumaas kasi yung calcium sa loob ng cell, ng cytosol, magbabind yan sa calmodulin. And then, pagdating nyo sa muscle lecture, matuturn on to, si myosin light chain kinase, which can eventually lead to muscle contraction. Yan. Another second messenger is cyclic GMP. Pag tumaas yan, basically, lahat ng A dun sa CAMP system, palitan mo lang ng G, no? Cyclic AMP, maging cyclic GMP. Adenylyl cyclase, gawin mong guanylyl cyclase. Yan. Pag tumaas si cyclic GMP, relaxation. And then, pabilisin ko lang ha. Yung G-alpha na yan, minsan nag-o-open rin ng mga channels. Okay? Ito, wala. In-emphasize ko lang kanina. Si CAMP ang mag-activate sa protein kinase A. Kinase will phosphorylate stuff. Cyclic GMP utilized by certain hormones or substances. Nitric oxide. Sino pa? Si BNP, brain natriuretic peptide. Si atrial natriuretic peptide. And ito is also known as endothelium-derived relaxing factor. Si BAN. Okay? Si BAN, they utilize cyclic GMP as a second messenger. Nitric oxide, mag-upo siya sa kanyang receptor. Okay, the receptor will be activated. And then, ito turn on ni receptor or yung subunit na yon, ito turn on si guanylyl cyclase o letter C. Letter C din yung lulutuin. Okay, cyclic GMP. P yung next, P yung i-activate next. Protein kinase G ang mata-turn on hanggang sa masunod kung ano man yung gusto ni nitric oxide or whatever hormone. Just remember, sinong gumagamit ng cyclic GMP system? It's ANP, nitric oxide, and BNP. Brain natriuretic peptide. Galing sa ventricle ha, hindi sa brain. This is atrial natriuretic peptide. Galing sa atria ng heart na nagpo-promote ng natriuresis. Natriuresis. Pampaihi. Dami ng ihi. Ang dami ng sodium dyan. Naiihi. Uresis. Ihi. Natrium. Natrium kasi si sodium yung Latin word. Wala lang. Ayun si cyclic GMP. Nitric oxide. Ano yan di ba? Lumulusot, gas. So, mag-upo siya dito sa kanyang receptor sa loob. Soluble guanylyl cyclase. Itong effector na yan, luluto ng second messenger, activate si protein kinase, magpa-phosphorylate, etc. Hanggang masunod ang gusto ni nitric oxide, which is smooth muscle relaxation para mag-dilate ang blood vessels. Catalytic receptors, from the name itself, receptor na nag-act as catalyst or enzyme. Okay, 2-in-1 siya. Example niyan, si guanylyl cyclase, utilized by this hormone. Ano pa? Receptor tyrosine kinase. Yung receptor mo, yung upuan mo, 2-in-1. May tyrosine kinase enzyme. Kinase, tyrosine. Yung kinase na yan, pupukpukin yung sarili niyang tyrosine residues. Yan. Sino gumagamit niyan, Doc? Yung mga pampalaki. Insulin, insulin-like growth factor 1. Ito kasi, it promotes lipogenesis, glycogenesis. Basta lalaki ka. Paano niya ipopromote yun? Biochem na yan. Okay? Ito si insulin, no? insulin stimulus for secretion is high blood glucose. Pag mataas ang glucose mo sa dugo, pancreas will secrete insulin. Insulin will do everything to let glucose come into the cell para hindi siya mataas ang sugar sa blood, no? Kailangan may pasok sa cell. Paano niya nagagawa yun? Kita mo yan, umupo siya dito. This is its receptor. And ang receptor ni insulin ay may tyrosine kinase. Okay? Pag upo niya dyan, may tyrosine residues to, pupukpukin niya yung sarili niya. That's autophosphorylation. Pag may phosphate na yan, may mga maiinggit na molecules, lalapit, magpapapukpok rin, mga IRS. And yun na. Kakausapin na yung loob ng cell mo. Oh, luto tayo nito, ng GLUT4 para makapasok si glucose. Luto tayo ng enzymes for protein synthesis, fat synthesis, glycogenesis, and glycolysis. Yan, mga gusto ni insulin. Ayun, nakita mo yung kidlat na yun. Yun yung pinag-uusapan natin. Okay, paano niya kinausap yan? Yan, tyrosine kinase chu-chu siya dyan. Okay? So, umupo si ligand, umupo si insulin sa alpha subunit na nice to know muna. Sa endocrine nyo pa malalaman to. And then, na-activate si receptor, pinukpok niya yung sarili niya, phospholipase niya, and etc. Kung naman gusto ni insulin. Tyrosine kinase associated receptor. Ano pa? May isa pa, yung Janus family of receptor associated tyrosine kinase. Yung JAK-STAT na sinasabi. JAK is Janus kinase. STAT is signal transducer and activator of transcription. Ito naman, ginagamit ng mga pampagrow, growth hormone, erythropoietin. Ito pampagrow ng red blood cell mo. Wala lang si JAK. Kasi JAK-STAT pathway. Ang dami naman, Doc. Don't worry, babalikan nyo to sa biochem. Utilized by cytokine as well. Wala, may pangalan lang yung ano niya, may JAK and may STAT. So, this is hormone na ng fat. Umupo siya sa receptor niya na JAK-STAT. Pag upo, pinosphorylate si JAK. O, tapos, nainggit si STAT. Ako rin. Papukpok rin ako. Paphosphorylate din. Then, na-phosphorylate yung mga STAT. STAT. Nag-couple silang dalawa. Nag-dimerize. Then, let's go together sa nucleus. Kausapin natin si DNA. Paluto tayo ng kung ano-anong protein. Okay? So, ganun ang JAK-STAT. Right? Just know na yung growth hormone utilizes JAK-STAT. Pati si leptin ha, JAK-STAT rin. May video for this. So, yun yung receptor na may JAK. Si insulin wala eh. This is utilized by cytokines. Cytokines, pang-utos ng mga cell gumalaw, whatever. Cytokine, nag-bind sa receptor niya. Si JAK, kinase yan eh. Pupukpukin niya yung sarili. Mag-phosphorylate siya. Sa mga Y is your tyrosine. Pagdating sa amino acids, it's tyrosine. Nainggit si STAT, ang kami rin, papukpok din, paphosphorylate. Natanggal yung mic. And then, STAT, let's go together sa nucleus. Yan, let's go together. Kausapin natin si DNA para masunod ang gusto ng cytokines. Yun lang. Mga areas na yan, nice to know. Alamin mo lang, cytokine, leptin, JAK-STAT ang ginagamit. Insulin, tyrosine kinase ang ginagamit. This is utilized by sino-sino? Growth hormone, prolactin, pampagrow ng boobs. Ano pa? Erythropoietin, pampagrow ng boobs. No? For milk synthesis to. Ito for red blood cell. And cytokines. Ayun namin kailangan nyo dyan. Again, may dimerization na nagaganap. Wala lang. More of biochem to eh. Then, lumilipat sila together sa nucleus. Steroid, thyroid. Tandaan, mga non-polar yan. Pupunta diretso sa lusot ng cell membrane and then bind to its receptor found in the cytosol. Okay? And then, let's go together. Hormone receptor complex. Punta tayo sa DNA. Pagpaluto tayo. Yan. Si steroid hormones mo, example nito ay cortisol, aldosterone, ano pa, sex hormones, mga estrogen, progesterone, testosterone. Thyroid hormone, ito, ang receptor niya ay nasa nucleus. Okay? So, sino yung mga lumulusot? It's DATs. They are DATs. Vitamin D, retinoic, or vitamin A mo, thyroid hormone, and steroid. Yan. Walang second messenger chuchu kasi sila mismo ang kakausap sa nucleus. Magsasabi doon kung anong gusto nila. Kita mo yan, inflamed do. Oh, yung binibigay usually pag inflamed, mga steroids, mga synthetic glucocorticoids such as clobetasol. Yung clobetasol na yan, mabagal mag-act. Kaya nga pag-apply, hindi ka kagad gagaling. Kasi, ganito mechanism niya. Pumapasok muna siya, then, kakausapin niya si DNA, aantay ka magluto ng protein. Walang second messenger chuchu, walang amplification na nagaganap. Masyado, compared dun sa mga polar hormones. Okay? So, memorize this para sa exams nyo. High yield to eh. Sino gumagamit ng cyclic AMP as second messenger? IP3, CGMP, tyrosine kinase, etc. Yan. For instance, cyclic AMP, wag mo na memorize. Ito, madali lang, oh. Oh, ito ang mnemonic dyan, GOAT. G-O-A-T. Oh, HAG. They utilize IP3 and DAG as second messenger. Okay, upo sila sa GQ protein. Yan. Or GQ receptor. GQ activates phospholipase C. Phospholipase C, magluluto ng second messenger, IP3 and DAG. IP3, palabasin mo si calcium. DAG, activate mo si protein kinase C. Sino gumagamit? Ito. Sino nang activate? Itong mga to. GOAT. Cyclic GMP. This is utilized by bad grandpa. Oh, si BNP, ANP, and nitric oxide. Okay? Or endothelium-derived relaxing factor. Wala siyang transducer. Dito siya mismo mag-effect sa guanylyl cyclase. Papaluto ng cyclic GMP. So, cyclic GMP ang second messenger ng mga to. Okay? Ang secondary effector, i-activate ni cyclic GMP is protein kinase G. Tyrosine kinase, yung mga pampagrow yan. Pag narinig nyo, tyrosine kinase. Who utilizes tyrosine kinase or which hormones? Insulin, pampagrow yan ng protein and fat and glycogen. Insulin-like growth factor 1, growth hormone, prolactin, PDGF. Yan. Ito, nice to know. Wag na yan. Cyclic AMP, all the rest. Kaya nga hindi ko pinapamemorize sa inyo eh. Anong second messenger? Or, ito yung effector. Ito yung gumawa sa cyclic AMP. Cyclic AMP will then activate this one, protein kinase A. Okay? So, ito yung mnemonics. Cyclic AMP, sino gumagamit niyan as second messenger? Etong mga to, na hindi ko minamemorize. Cyclic GMP, yan, i-memorize mo. Konti nila eh. B, A, and E. Brain natriuretic peptide, atrial natriuretic peptide, and endothelium-derived relaxing factor. Kailangan ba i-memorize, Doc? Hindi na. Mag-abbreviation na lang tayo. Okay? Yung mga hormones na yan, utilize cyclic GMP as second messenger. Ito yung sinasabi kong GOAT. Pag sinabi ko, oxytocin, anong second messenger niya? IP3 po. And DAG. Ha? Ano pa? Correction, TRH, thyrotropin-releasing hormone, IP3 din. Yan. Usually, mga releasing hormone to, gonadotropin-releasing hormone, hindi pala yung correction kasi tama naman sinabi ko kanina. Ang hindi lang, ang releasing hormone lang na hindi IP3 ay si corticotropin-releasing hormone. Okay? All the rest, pag galing sa hypothalamus, mga releasing hormones na yan, IP3 ang second messenger. Okay? Intracellular, lumulusot. Oh, ito yung sinabi natin, vitamin D, vitamin A or retinoic, mga sex hormones yung mga yan, and then T3, T4. Receptor tyrosine kinase, yung mga pampagrow. Okay? Pampagrow, growth factor, whatever. Insulin will make you big rin eh, so pampagrow yan. Non-receptor tyrosine kinase, si EPO, ito yung mga kanina sa JAK-STAT pathway, growth hormone, cytokines, leptin, yung mga hindi ko in-emphasize, hindi ko tatanong sa exam. That's it. Thank you for listening. If you listened well, sana mag-notes ka and good luck sa exams. Thank you.